Thank you, Jed. May I ask the speakers from the morning to come to the podium, to the desk? And then, oh, wonderful. So we have Roy, Jed, and Mark coming up. Perfect. Okay, so we do have all the speakers now. I know you are eager to ask questions. 1 minute is that the questions can be asked, of course, from live questions from the podium, from the audience, but also please put your questions in the chat box. And we are monitoring the chat box. We'll answer those as well. Roy, would you like to take on the discussion since you are able to join us?
Yeah, thanks, Hedy. What a great session and great meeting. And I am monitoring the chat. I don't see anything in the chat yet, but if it comes, I will let you know.
Okay, then shall we say, go ahead. We have people standing. We have people standing at the microphone. Go ahead.
No, maybe as we start, while people come to the microphones, I can't see the microphones there. I'll just throw to the panel. So we want to have more success in phase 3. Alex really showed us that so well. What would be the critical thing to take something from early phase to the next phase of trial? Is it phase 2 results? Is it what's the best endpoint? Do we need to have a biomarker? I think the question I would ask the group is, how critical is a biomarker? Some sort of and we just heard a very nice talk from Jed. And I don't know if anyone wants to start with that.
Well, every speaker actually mentioned biomarker, either how to validate. Elhad said don't use too early. Keith said importance of multiple biomarker and not one, so maybe one at a time. Keith, would you like to start since you mentioned that as well?
Yeah, I mean, Roy, you're asking about a response biomarker, yeah? I mean, I think
right,
in terms of level of evidence.
The group.
Right. So I mean, I guess I didn't talk about this very much. Well, okay. So I did mention the imaging piece, and maybe what I would suggest is that we have reason to believe that things like greater increase of CD8s into tumors following a therapy. I shouldn't say investigational because it's really benchmarked on PD-1, and circulating tumor DNA, quote unquote, response. Again, credentialed with lots of retrospective PD-1 data, that those are, those prove to associate with clinical benefit. I'm not looking at any FDA colleague here when I say this, but referring more to kind of pre-regulatory consideration. So if we're moving from an early phase trial to later phase trial, I would say we learned on the back of PD-1, those types of associations. I think for any other type of biomarker that's more mechanism related, so related to an investigational therapy, I'll make the point that I always make, which is that unvalidated drugs have unvalidated biomarkers, right? So like being able to show that, something that depletes MDSCs, that's evidence of benefit or reasonably likely to predict benefit, I would say we don't know that until such time as we get the clinical outcome data and then can look back and say, oh yeah, it either did or didn't. So again, I think where we are as a field right now is we've got these learnings from PD-1 and biomarkers that in the case of some novel imaging approaches are related to an immune mechanism. In the case of ctDNA, not. Those are the types of biomarkers, response biomarkers, that I think we're possibly as a field positioned to take risk on, but risk, right? I mean, we risk that a novel therapy that comes along and moves the needle in terms of an imaging endpoint or deeper circulating tumor DNA response. We don't know until we get there that actually associates with clinical benefit. So, I'll finish by saying I absolutely positively think we are at a point in the field where some degree of risk taking is needed. I meant to emphasize mostly that we need to really rely more on early trial incorporation of pharmacodynamic biomarkers to be able to really confidently support further clinical investigation before even doing a randomized phase 2 trial, to be clear. To me, randomized phase 2 trial is like, that's a long road, big commitment, and one that small venture-backed biotech companies are not willing to make to be totally clear. So got to come up with strategies that are short of that, in my view.
Elhad, you were nothing.
Oh no, I totally agree. I think that the truth is these response rate biomarkers like ctDNA, certainly the imaging advances that have been discussed at this meeting and in other venues are extremely interesting, but they are essentially just surrogates at this point until they're actually validated in the sense that they correlate with something that we consider clinically meaningful. It's very difficult to say that is necessarily going to cause a given advance. They're extremely promising, but I think that probably Dr. Flaherty would also acknowledge, there are questions in ctDNA, whether a tumor-informed or agnostic ctDNA assay is something that we can potentially rely on, and whether or not in every tumor type this is something that we can potentially rely on. There are a lot of criticisms of RESIST, which I think we're very familiar with in the oncology sphere. However, the reason why RESIST is still there is probably because it's difficult to prove that something's better. And it'll take some time to get to that point. And even RESIST itself is merely a surrogate. Everything that we're talking about with regard to response rate efficacy is really just looking at surrogates. We know what patient benefit is in most cases, and that's what we are trying to emulate.
So you want to, of course, Alexandra.
I would answer your question, Roy, by categorizing the biomarkers we need into three spaces. Number one, are you treating the right patient to try and get a meaningful effect size? And that may require a selection biomarker. Number two, are you modulating the biology you want to modulate? And that may require an on-treatment biomarker. And number three, are you actually helping patients? Of course, in the registrational setting, you need RECIST or a longer endpoint, but in a phase 1 study, you can certainly use things like tumor size change and ctDNA to get a signal. And so I think the answer to those three questions is a little bit trial specific, but that's sort of how I conceive of where we need to use biomarkers to get to proof of concept sooner and with fewer patients.
So, Jed, I can see on your face dying to answer. Go ahead.
You have very good vision, Hetty. So I couldn't agree more with what's been said, and especially Alexandra's summary. And I really think that from my viewpoint of the presentation that I gave, that the important change that I would suggest is that we really rethink the way we judge the outcomes from phase 1 clinical trials. And I can't stress enough the fact that, before abandoning an entire class of drugs, we need to understand whether those drugs caused the expected change in the immune response when we're talking about immunotherapies that was predicted based upon the preclinical data. And once we have achieved a yes or no answer to that question, then we can ask, well, how then does the challenge of patient benefit become best addressed? And then once we have a mechanistic biomarker proving that drug hits target, then we can go on to say, well, for which patients is this change most relevant? So, if we're talking about an agent that depletes myeloid-derived suppressor cells or repolarizing them, what changes in the peripheral blood or the tumor microenvironment are seen? Which patients have a high number of these cells to begin with? And then and only then can we think about, the most important outcome, which is really patients living better as a result of this intervention. So I think that because of the quote-unquote go-go nature of this field, for all the right reasons that we're trying to help patients faster, we've expected that the next PD blocking drug is going to come along any minute now, and in phase 1 trials is going to cause tumors to shrink. That, I hope that happens, but it may not. And so we need to really refocus on what we expect to learn from these small early phase 1 trials. Have a little bit of patience for the data to mature and for us to understand the best clinical venue in which to use these drugs.
Thank you. Roy, I have 5 people in the room standing, eager
Okay,
to
go
ask
for it.
questions. Go ahead.
Thanks. This is Samir. Jed, I would like to just follow up on your discussion, because we had some discussion yesterday, and I cannot agree more. And you and I had this discussion before also, that rushing to ditch a whole class of drugs is kind of like sad, because we lose lots of patients and lots of money and for no reason. Now, the question that I have, though, you take any of those drugs like Gitter, like OX40, we identify the mechanism, we go to phase 1, we found that it is reproducible and the mechanism of action or proof of mechanism has been accomplished. The question then becomes how to move this from biologically active drug, because it's still not active from a tumor perspective, to a more positive outcome. And that has 2 paths, right? One is as a single agent, and second as potential combination. So if we take the combination and we go back to the lab and do the combination and found that this particular combination would work, is that enough? Or do we have to go to resistant models and find why it doesn't work and whether there will be any impediments to take it forward? That's one question. The second question is, what would be the path still as a single agent? Because it might still work as a single agent, but we don't know exactly how to move it from one step to the next. And this is a big part of this conference. This is why I'm just dwelling on it and trying to get your thoughts on this.
Jed, that was for Jed, right? Yeah. Jed?
Yeah, so Samir, that's a great summary. And I think that, step 1, once you have the mechanistic biomarker established, right, then you can take the next step with a predictive biomarker and identify that patient population. So I think that sequence of events is correct. I think that, how do you ultimately, what does success ultimately look like? I would actually open that to Elad and also to Mark as well for their insights about how do you design a clinical trial that incorporates that, because we recognize that it's going to be a combination strategy. So do you need a contribution of components assessment? And you very well may. Can you use a patient population where the patient represents their own control? So if you believe that the new agent somehow works in a complementary manner with say, for example, a PD-1 or PD pathway blocking drug, if you look at a patient who has just progressed on a regimen containing a PD-1 drug, turning around that progression on an individual patient basis, how realistic is that as a registration endpoint? We have discussions like this quite frequently, and I think that this kind of venue with multiple stakeholder groups represented would be great to hear from all sides.
So Jed, that's really fantastic, because this is really a segue. You read my mind. I was taking this and pose it to Mark, because he gave that talk, and to Elad. Because Mark and Elad, you mentioned always the issue of randomization. And what Jed mentioned is exactly what I was going to put forward. Do we need always randomization, or can we move on a rescue basis for the failures and say, well, that might be at least a platform for an accelerated approval, or there is a path for this moving forward?
Can we ask Mark to answer? I'm sorry to be the bad guy, but we only have 6 more minutes for the discussion. Mark, go ahead.
Yeah, so I just want to comment briefly on the response biomarker kind of theme as well to start this off. And one of the things that we had discussed yesterday and today as well is thinking about how biomarkers are used in a clinical trial, and think about the risks that are associated with its use. And when thinking about the different ways biomarkers could be utilized in a clinical trial, there's use of biomarkers for prognostication, for example, to identify patients who may be more likely to have, a particular outcome. And that may be incorporated into, stratification. So the trial population isn't predicated on that result, but the trial population would be stratified for randomization to predictive biomarkers. So incorporating that information into the clinical trial for patient selection and requiring that the patient has had a particular result to enroll. And then to the point of response biomarkers, which is a very key point, the issue about how is it being used, when is it being used in a clinical trial. Early on, that response / pharmacodynamic biomarker could be used for target engagement, could be used for pharmacodynamic assessment for dose selection of critical component, which we actually haven't really discussed in this conference, but think about dose, an optimized dose for the combination, to then incorporation as potential candidate surrogate endpoints, then as a surrogate endpoint that would be reasonably likely to predict clinical benefit, thinking about accelerated approval, to one that's a validated surrogate. And all along that kind of path and how it can be used in the clinical trial, the risks of an incorrect conclusion based on that result increase. And that's really a risk to patients. So you can think about, let's just use overall response rate as an example, as a pharmacodynamic response endpoint. Of all of the PD-1 inhibitor approvals, approximately 40% or so were based on response rate. Having that endpoint as an endpoint reasonably likely to predict clinical benefit has really, accelerated, bringing promising therapies to patients ultimately. What is that next generation pharmacodynamic response endpoint that could be utilized? And then I think it was mentioned earlier, what information do we know about in terms of its correlates with something that is, clinically meaningful? So along that paradigm, you could think about, depending about what is known in the entire development program, or the associations of that biomarker with important clinical outcomes, could be utilized for contribution of components. And we have, and I'll just finish and say that we have been very transparent, that an earlier endpoint in a clinical trial, such as response rate, may be adequate to demonstrate the contribution of components. But randomization is a very important concept, Samir. Thinking about reducing the known unknowns as well as the known, the unknown unknowns in terms of how patients do and making kind of conclusions, about contribution.
Thank you. The question from the podium.
Yeah, hi. So two big
Audience.
themes we've learned today and yesterday are, haste can sometimes make waste. If we go too quickly, that can be successful, but that can also lead to an unacceptable rate of failures. And then the second thing is how to learn from these failures. I think, Alex, you mentioned, how to incentivize and learn more from some of these case studies we've learned. Do you guys have any thoughts on incentives for, how to take deeper dives into the pathways, the studies that didn't succeed, so we're not just throwing the baby out with the bathwater?
Alexandra, that's your quote.
Thanks, Gideon. I think it's a real challenge. I think to answer the question, we have to answer, well, why aren't these analyses being done? And quite frankly, I think expense is a big part of it. In other cases, there aren't sufficient samples to make sense of and those sorts of things. But for the large phase 3 studies, it really is an issue of time and money. So if we could either align those, for example, if there were to be cases like if you look back at the development of EGFR inhibitors where such retrospective analyses not only rescued the drugs but turned them into incredibly successful drugs by defining who the benefiting patients are. I think successes like that can engender dedication to such translational work. Another option would be to require it, but I think that will take a whole nother discussion. I see Roy has his hand up.
So, Roy?
Yeah, John Mendelsohn used to tell us from this podium that thousands of patients were treated with EGFR inhibitors before we knew about the mutation. I'd say that the public-private partnership model is a good one in that the company is working with academia, working with community. There are tons of samples there. And one thing that I would push on top of what Alex just said, we have to look at these samples and understand response, stability of disease. There are so many better ways now to look at the radiology, at the ctDNA, and understand what some of the markers are and learn even from trials that are not positive. And I can tell you, even in the lung map experience, there are 4,000 specimens, and they're still available. We can't just bank them. We have to use them and use them to figure out mechanistically what the next markers will be.
Right. Elhad, I saw you wanted to say something. No?
I just thought to mention something also that Dr. Herbst mentioned earlier. There's the CMAX, were supported also by industry, the Partnership for Accelerating Clinical Trials. And there was actually a pathway for industry trials to provide samples. Now that has now at this point closed. I think the PACT partners with FNIH are still considering whether or not they're going to participate in the next version of CMAX. NCI has already committed, and so we are actually funding the CMAX going forward. But that would certainly be a method and potentially a way to incentivize companies, because essentially it would be provided for by PACT resources, which ultimately come from industry. But I acknowledge there's really a problem. I don't think that it's something that's so simple to deal with, in the sense that we're talking about private loss, as it were, from an individual biotech or pharma, and public benefit for the rest of the community, and that is always hard to push forward, even if we do de-risk it and lower the cost by providing that kind of subsidy or collaboration.
So, Erin and Cheryl, I'll just take the two questions. Run a little late. Go ahead. No, go ahead.
My question is actually quite related. I'm Kim Rathmel from Vanderbilt. It feels like we're still in the discovery phase for biomarkers, and so having access to these kinds of samples and reagents. What I was going to ask was, are there I won't say standards, but guidelines that need to go around what kinds of samples get collected that are then accessible as a resource for use in the future?
So who would like to answer that? What resources available?
So I'll at least just mention that NCI trials do end up being something of a public resource in a sense, and certainly the NCTN trials, the National Clinical Trials Network trials, they can, subsequent to publication, you can apply to access those resources. Absent, of course, things like PACT, however, I don't think that there, or the CMAX, these Cancer Immune Monitoring and Analysis Centers, I can't say that we're necessarily providing funding up front for an effort, but certainly one can at least see what's available through our correlative science committees and apply to receive these resources that we consider ourselves a public steward for.
Roy?
Well, in the lung map, through the NCTN, these samples should be available publicly. There's a translational research committee, there's a governance, and there's a process. But my feeling is that these samples are underused. And I think that this is something we need to promote more of policy-wise, to use samples that are banked. We're good at banking, but we have to learn to lend. So that's something we're actually trying to promote right now, because without finding the next generation of markers, I think we're going to have a very slow slope.
Our last question. Go ahead.
I've been UC San Diego for Alex. So related question of biomarker testing, in the context of head and neck cancer, until very recently, the big pivotal trials were done through pharma and not through NCI cooperative groups. So, as you know, in the case of what we talked about yesterday with Chris, the 9P21 story was proposed to you through LOIs and went through mechanisms and things. What do you think the process is to do that, to take it from kind of these real world cohorts to look at completed key trials like Keynote 40, for instance, to get the baseline samples for exome sequencing to look for resistance or prognostic markers, select patients who may do better, and potentially to rescue drugs like the Pfizer Javelin drug, which, quote unquote, didn't work, but if in a de-identified way you identified a resistance marker and there was a signal in patients that didn't have that, it could be useful for drug development. So just wonder what your thoughts were, Alex.
Thanks, Scott. I think it's a great question. So my experience in big pharma is limited to Merck, as you know, where there's a collaborative program where if investigators want to look at a certain biomarker, they can apply to do so retrospectively. It seems like the different companies have different approaches to this. I do think pretty uniformly companies don't give out their physical samples, but there are pathways to examine such questions retrospectively. There are some regulations that make this trickier. So there are some governments that don't allow germline sequences to be looked at or shared, and that can complicate matters. I think the real challenge lies in taking those retrospective findings and translating them prospectively, because many times you can't look at those things until a drug has been registered around the world, which takes often a year plus after it's been registered in the US. And so by the time the drug's registered everywhere and then you've done the analyses, the space has moved on to some extent, and so there isn't a study to which that finding can be applied prospectively. I haven't seen a situation quite as yet where that's been solved. That being said, if any of you went to SITC on Wednesday, there were 2 great talks by Genentech and AstraZeneca demonstrating sort of their approach to the translational space, which I think suggested that they are moving towards doing the sort of thing that you're suggesting.
Yeah, and just quick, you said sending samples out, but how do you feel about sending de-identified whole exome sequencing sample data out as opposed to samples from a pharma point of view?
My impression is that's quite rarely done because once such things have left, one can't guarantee that they're being handled in accordance with the regulations with which they were collected.
So thank you. Any closing remarks from our speakers, because our discussion was short? And Jed, short. Anything you would like to say?
No, I'm just very grateful for the opportunity to participate in this important discussion.
Thanks, Mark. What did you learn?
No, nothing additional. Thank you for including me in this discussion.
Thanks. Keith?
No, too many things to say to fit into closing remarks.
I'll add.
Yeah, I think that I'll be available, obviously, at this meeting, but also really open to have any further discussions on these topics. It's extremely, obviously, intellectually interesting and stimulating, and we have to work together to find paths forward for our patients.
Alexandra?
Agreed, and I find it heartening that we have gatherings like this, because I think only through such cross-functional work will we move forward.
And now from Roy, our comrade, the final words.
No, I just think, we've seen such progress in all these tumor types, and many think that just adding drugs together will be the next step, and it's not. There's a very nice paper by Mike Atkins and the SITC looking at phase 2 to phase 3 and what the criteria should be that I would recommend to people. And everything we're talking about here, we have to add science to our trials and learn rather than thinking that we can just, see the next big leap just randomly. So I think this meeting's been tremendous, and thank you for including me virtually.
Thank you all very, very much. Sorry we are a little late. Thank you.
Why don't we take a 5-minute break and come back in 5 minutes?
Oh, good.